Okay, I want to welcome everybody back. And we have a very interesting also second session on disease models. And our first speaker is Arnold Han from Columbia University in New York. And the talk is entitled Mechanistic Insights on Celiac Disease from Celiac Disease Patients.
Great. Thanks so much for the invitation here and the opportunity to speak. I am from Columbia University, and I'm an immunologist that studies celiac disease, but I also have the privilege of actually seeing celiac disease patients in my clinic. And I know this is about models of celiac disease, and I'm going to primarily talk about a really important model that we study, is the model of human celiac disease in humans. So this slide I really don't need for this audience, but we all know that celiac disease is a very common autoimmune disease that affects about 1% of the population. We know that the celiac genes, so to speak, are HLA-DQ2 or DQ8, so they confer susceptibility to celiac disease. But importantly, not everybody that has those HLA types does have celiac disease. Now, we know that celiac patients do share many similarities in their immune system with other autoimmune diseases and are more susceptible to these other autoimmune diseases. And, you know, this is something that's unique to celiac disease among all T-cell mediated autoimmune diseases, is that we actually know the antigen trigger to celiac disease. So it really gives us really unique opportunities to study immunology. So this is what we understand currently about celiac disease. And I don't know, do I use a pointer down here? No. Okay. I guess I don't have a pointer, but I'll figure it out. So we know that the CD8 T-cell is critical in celiac disease, right? So we know that dietary gluten is processed in the intestine and broken up and presented by antigen-presenting cells only on HLA-DQ2 or DQ8 to CD8 T-cells. Now, the CD8 T-cells are well appreciated to be important drivers of the disease. Obviously, this disease only occurs in HLA-DQ2 or DQ8 people. And these T cells, we understand, are not only responsible for initiating inflammation in celiac disease or recognizing that trigger, but also producing these antibodies that we track in terms of disease activity, antibodies against tissue transglutaminase, which is a self-antigen, as well as gluten itself, okay? But we also have long understood that CD8 T-cell immunity, although necessary for the disease, is actually insufficient for the disease. And we know, we've appreciated now for quite some time, that the cells actually damaging the intestine or responsible for killing the intestinal tissue are in fact different types of T-cells. So not CD8 T-cells, but IELs that are either CD8 or gamma delta T-cells. And these cells have a really distinct phenotype in that they express NK receptors that are used by natural killer cells. So they have like these innate-like immune properties, and they're responsible for killing the tissue. Now, what's been a long-standing question in the field is, okay, so we know that CD8 T-cell recognition of gluten is essential for the disease and appears to initiate the disease, but how did this in turn lead to damage by other types of T-cells that are not CD8 T-cells? And really the model that people really bought into for quite a few years now is that CD8 T-cells first trigger inflammation, and then this inflammation in turn later triggers these IELs that are sitting in the intestine. So why is this such an important question? Well, you know, like celiac disease, I think most autoimmune diseases, if you look at this chart, are associated with class 2 alleles. So celiac disease is associated with DQ2 or DQ8, but these autoimmune diseases tend to be associated with class 2 alleles. Now, class 2 MHC is responsible for activating CD8 T-cells, right? And in most of these autoimmune diseases, though, just like celiac disease, the end effector cells that are actually damaging the tissue are not CD8 T-cells. So I think that this question of how a CD4-driven autoimmune response leads to tissue damage by other types of T-cells is really important. It's going to hold true and be important in the understanding of these other autoimmune diseases. Now, I guess my title, which I forgot to edit, but it's a more provocative question, right? Because since we don't know the antigens that actually trigger these other autoimmune diseases, one question is, could they possibly be exogenous antigens, right? So for instance, we focus on the pancreas and diabetes. Is it possible that the antigen that actually initiates diabetes doesn't derive from the pancreas at all? So this is a paper I published now, well, it's been a long time, over 10 years ago. And we found something pretty interesting. So we've heard about this gluten challenge where, you have a celiac patient on a gluten-free diet. They voluntarily eat gluten for a short period of time. And it's been reported, and I'm going to direct you to panels on the right, by Bob Anderson and others and Ludwig Solid, where, at 6 days, a very interesting phenomenon occurs. You see this really big upregulation, CD4 day 6, of gluten-specific CD4 T-cells circulating in the blood. Okay, and this has been known for quite some time, and it's garnered some interest as a diagnostic tool. But when I was in Mark Davis's lab as a postdoc at Stanford, we discovered something unexpected. And that thing unexpected was that not only do we see CD4 T-cells proliferating in the peripheral blood and circulating, we also saw this really big upregulation of CD8s and gamma delta T-cells that really look very much like IELs, these IELs that we think are damaging the tissue. Now, why was that surprising? Well, as I alluded to, the existing model suggested that first you had CD4 T-cell inflammation, which in turn triggered these IELs later on, right? And to us, this implied, at least when I first looked at this data, this was unexpected because it seemed to go against that existing model where you first get CD4 activation and subsequently get IEL activation. And this seemed to imply there was something interesting happening where you're getting activation of IELs and CD4s at the same time by dietary gluten. And another reason why this is really interesting as an immunologist is that, despite many years of research, nobody has shown that these IELs actually recognize gluten themselves. All right, so the question is, then how are you getting such rapid activation of CD8s and gamma delta T cells, IELs, that don't recognize gluten by dietary gluten, right? And this was somewhat controversial when we proposed this model, and it was kind of discounted as some sort of, non-specific recirculation, which does occur in various intestinal immune conditions. So we kind of, I still like this model a lot, but it wasn't widely accepted by the field, okay? So here we go. Fast forward to when I became a faculty member at Columbia. We started looking at human celiac disease. Now, we all know how important celiac disease is as its own entity, but it also provides a lot of interesting tools to just understand immunology, right? So we have, because we know the antigen, we have patients with many different stages or different distinct stages of disease. So not only do we have healthy controls and also people with active celiac disease, but we also have patients who have celiac disease but are on a gluten-free diet. And by all metrics, their disease really kind of goes away. Their intestinal biopsies pretty much normalized, their antibodies normalized. So these are people with inactive disease. And we have this also patients that are refractory to gluten-free diet. And we also have this gluten challenge, mechanism which I also told you about. And interestingly, we also have this potential celiac disease population, which I'm going to talk a little bit more about. But not only do we have these unique different states of disease, but we also can access the tissue quite easily in the form of intestinal biopsy. Now, I'm going to talk to you about, so what we've done is, I should go to this slide first. I'm going to talk to you a little bit about potential celiac disease because it is pretty interesting in our data. So potential celiac disease, again, is another very unique aspect of celiac disease that gives us a really important tool, I think, to understand celiac disease. It occurs in about 20% of new celiac diagnoses. It is a CD4-mediated phenomenon because it only occurs in DQ2 or DQ8 individuals. So this seems to imply that those CD4 T cells are getting activated. You do have positive serology. So you have anti-TTG and anti-gliadin antibodies, just like a celiac patient. So the CD4 response seems to be intact. You're making the antibodies, you're activating those CD4 T cells, but there's no intestinal damage. Okay, so there's some break between this chain of CD4 activation and intestinal damage, right? And so this is a patient population we were very interested in and we studied. And some of these patients progress to celiac disease, but others may remain disease-free for quite a long time. So our approach was we started just looking at observing our patients, okay? So we took intestinal biopsies and looked at the T cells from those biopsies with many different parameters. We did single-cell RNA sequencing on those T cells. We looked at 200 cell surface markers by CytSeq, and importantly, we also looked at the T cell antigen receptor. So this is the kind of picture we get. We get a landscape of duodenal T cells. And these are just UMAP plots for those of you who aren't familiar with these. These are just looking at the T cells in the intestine, looking at the diversity of those T cells in two-dimensional space. Okay, so these are different, each cluster represents different types of T cells. And so here's a vignette. We can show that gluten does induce CD4 T cells that has been described, and we appreciate now, to recognize gluten. So these are these TFH cells that have this distinctive phenotype, kind of look like effector CD4 T cells with a T follicular helper phenotype. And we observed that those go up in active celiac disease and pretty much go almost back to baseline on a gluten-free diet. Interestingly, these patients I talked about with potential celiac disease do mount these CD4 T cell responses, right? Now, another thing that we observed is that in addition to these T follicular helper cells, we also upregulate Tregs, which seem to have an anti-inflammatory phenotype. So this is something we commonly see in biology and immunology, where you trigger a positive stimulus, and you also at the same time trigger a regulatory stimulus, okay? And there's been some sort of controversy in this field whether these Tregs are indeed recognizing gluten as well. And our data seems to suggest that they are not, because they do not really share TCR repertoire with these TFH cells, nor do they have these canonical gluten recognition motifs. All right, so I'm going to focus most of this talk on these IELs that we talked about. Remember, these are cells that we think are actually killing the tissue in celiac disease. And there are two clusters of IELs that we see in patients, okay? Now, the cluster on the left, which is circled, consists of gamma delta and CD8 T cells. Now, in most patients without celiac disease, most of the intestinal epithelial lymphocytes are going to look like the ones on the right, the cluster on the right that is not circled, okay? And we would have very few, people without celiac disease would have very few of these T cells on the left cluster that's circled, and most of the T cells in healthy individuals would be gamma delta T. Of these types of T cells would be gamma delta T cells. But in celiac patients, that cluster on the left, which consists of CD8 and gamma delta T cells, is really expanded and really is modulated by the states of disease. Now, what do those T cells, or what is the phenotype of those T cells? Well, they express markers consistent, very similar to natural killer cells. So they express these different. Do I have that on this slide? No, they express NK receptors and NK-like receptors, very similar to these type of cells called natural killer cells, okay? So the way you could look at them is they're kind of thought to be at the border between innate immunity and adaptive immunity. And what we know on the clusters on the left is they're very, profoundly modulated by disease states. So I'm going to point out three different phenotypes of those cells, okay? Natural memory 1, which is on the top, has this tissue repair promoting phenotype. And most healthy individuals, most of our T cells are natural TILs, look like that, okay? So they're not actually mediating inflammation. Now you go to the far right, these are natural effector TILs. These are the ones we think are actually damaging the tissue. They're highly cytotoxic, they express markers of cytotoxicity, and they're very pro-inflammatory and produce lots of interferon and granzymes. And sitting in the middle somewhere is this natural memory 2 population, which we think is an intermediate between the population on the top and the population at the bottom. Now, we did T cell receptor analysis, and they all share T cell receptor repertoire, indicating that there's plasticity between these populations. Now, what happens in disease? Well, not surprisingly, the natural effector T cells, the ones that we actually think are killing the tissue, right, is significantly upregulated in patients with active celiac disease in red, and really absent in controls, and pretty much absent in patients on a gluten-free diet, okay? The natural memory 1 population, which is on the top, is kind of interesting. In normal healthy individuals, predominantly those cells are gamma delta, but in celiac disease, you get an upregulation of those cells, even on a gluten-free diet, that are CD8 positive. Okay, so we think that these cells kind of represent the cells we see, they persist. So this is what Bana Jabri and others have referred to as this permanent scarring that occurs in celiac intestine, where if somebody has celiac disease and goes on a gluten-free diet, their immune cells never return completely to normal. And what happens is they develop an increase in these natural memory 1 cells that kind of persist. And what we think is happening is they're kind of a resident memory population where once you get exposed to gluten again, they're called into action really quite quickly, and they cause disease. Okay, so patients on a gluten-free diet predominantly have natural memory 1 cells, which persist pretty much for a long time. People have shown even decades. And when you get celiac, when you are actively consuming gluten, they shift over to this highly cytotoxic natural effector phenotype. Now, one thing that was interesting and somewhat unexpected here was that these potential celiac patients do seem to also upregulate this natural effector CD8 population. So they upregulate the cytotoxic phenotype population, even though they don't have intestinal damage. So the simplest explanation, given the model I had given you before, would have been that potential celiac disease patients just don't mount the IEL responses, but they mount the CD4 responses. And there's an uncoupling somewhere along the line. But what was a little surprising about our findings is that no, actually, those IEL populations, those cytotoxic IEL populations, are indeed mounted by potential celiac patients, even though they don't have any discernible intestinal damage. So I'm going to go back to this gluten challenge data. Now remember, I said that the gluten challenge data and the model we proposed over 10 years ago was somewhat controversial. People discounted it as, well, what you're just seeing is a recirculation of intestinal cells that occurs in inflammation. These aren't really celiac related. These are kind of bystanders. And so this is how we went about trying to prove our model, okay? So what we did, we took celiac patients who were on a gluten-free diet and they underwent biopsy. And then we subsequently, after they underwent biopsy, we subsequently gluten challenged them with a 3-day gluten challenge and harvested their T-cells at day 6. And so what this enabled us to do was actually match the T-cells in the intestine to the T-cells in the circulating blood by T-cell receptor sequencing. And so what are these circulating T-cells then? Well, predominantly, they're actually natural effector cells. So these are the cells that are highly cytotoxic and look like they're actually the ones that are killing the tissue. So you really rapidly mount these highly cytotoxic T cells that is occurring very fast with the exact same kinetics that you're actually stimulating the CD4 T cells. And what else did we learn about these T cells is that when we looked at their TCR repertoire, they look very similar to one another. Here's an example, the way I would explain this to non-immunologists. You know, this is an example of convergent evolution, right? So you look at the shark, the dolphin, and the puma, right? Genetically, phylogenetically, the dolphin and the puma have more in common because they're both mammals, but the shark and the dolphin have co-evolved many similar traits because they have to go swim in the water, right? So when we look at T-cell receptor repertoires, when we see similarity in T cells, it seems to imply a common selection mechanism. And we see many, many examples of this through these IELs. And what was interesting about that is, well, this seemed to imply to us that they're recognizing an antigen. But despite 30 years of work, nobody has shown they're recognizing gluten. So in fact, what is the antigen that they're recognizing? And this is an ongoing subject of investigation in our lab. To summarize this part, we think there's few things happening. So we understand a lot about what's happening with CD4 T cells. You're getting this activation of gluten-specific CD4 T cells. But what's also, I think, happening in parallel and not necessarily downstream is we're getting this rapid programming of natural memory 1 TILs, right, to natural effector cells. So maybe I'm going too fast and didn't make this as clear. The patients that underwent biopsy and then gluten challenge, what we observed is their natural memory 1 cells, which are anti-inflammatory in the intestine, within 6 days, boom, they become natural effector IELs with very, very cytotoxic phenotypes. So this seems to imply that, yes, it is a very deliberate, it is a very fast process that is occurring in parallel, but not necessarily downstream of these CD4 T cell responses, okay? So I'm going to talk a little briefly about what we're doing now, and this is mostly unpublished. Okay, we're in time. Okay, so I'm going to go through these really quickly. As I mentioned, there's a lot of unique tools we have in humans to study celiac disease, but there is a limit, as I'm sure Dr. Flavell will tell you about. You can't do experiments or actually do interventions on patients. So what we're using now in our lab is this air-liquid interface organoid culture. And there's a really nice paper in Nature that was published this year by Calvin Kuo's group, where we can take intestinal organoids from patients, and they retain their immune cells in them, and we can actually recapitulate celiac disease, as we show here. And this is an organoid where we can treat it with gliadin, and we can actually get intestinal inflammation that is mediated, appears, by CD45 positive T-cells, and we can actually block that from anti-MHC class 2 by blocking the activation of CD4 T-cells. All right? So I am going to stop here. This is kind of the questions we're trying to answer in the lab. We know that this reprogramming of IALs occurs. So what is the antigen? What are the factors that lead to that? We know that these natural effector cells occur in potential celiac patients. So what are the triggers then? What is the bottleneck in potential celiac disease? Why don't they have intestinal damage even though they're mounting these T cells? And we do think that these are actually epithelial factors based on our preliminary data that is actually mediating this, and we're investigating that as well. So with that, I'm going to end with the acknowledgments, which kind of disappeared. But thank you. Thank you for your time.
Thank you. Thank you very much. So now I'm going to ask Kati. So Kati Juuti-Uusitalo from Tampere University in Finland to talk. Her talk is entitled Personalized Small Intestinal Preclinical Model of Celiac Disease.
Thank you, Debs. So thank you for this opportunity to talk about preclinical model. Okay.
You'll have to press it when you to advance.
Okay, thank you. And then thank you for this opportunity to talk about the preclinical model of celiac disease intestine. So, okay, doesn't show there. So I guess that's this is the bread and butter for all of us about the celiac disease, how it's driven by the dietary gluten, rye barley prolymins, which are very resistant for the digestive enzymes within the gut. They have been modified by the transglutamases, which then deamidate the gluten, and it can be taken up by the T cells and then presented to the CD4 T cells, which then get activated and then start to produce the inflammatory cytokines, which then cause the inflammatory milieu to the small intestinal lamina propria. And then it alters the inflammatory status and morphology in the small intestinal epithelium. We all already know that there are certain genotype needed, which is the HLA2 DQ8, but then we still don't know that why only approximately 4% of the carriers develop the disease. Who are the others? Why are not those all celiac disease the predisposing gene carriers developing the disease? One reason might be the environment, and then also that the epithelial cells, they have been thought to be merely bystanders in this process. Just recently, Elena Verdu's laboratory showed that also the small intestinal epithelial cells are able to act as antigen-presenting cells. When then this is added to the quite recent knowledge that also epithelial cells can produce the tissue transglutaminase into the gut lumen, we actually have all the machinery for the antigen presentation. So the adaptive immune response is quite well known already, but what is the leap from the gluten till the adaptive immune response? It's not known, so that we are still missing some parts of the innate immunity reaction before that. And then to study that, we could do that, of course, in the human body, but then that is a bit cumbersome. So for that reason, we should have a preclinical intestinal celiac disease model. For that, we should have the CD genetics. So for that reason, the old-fashioned small intestinal models from the immortalized epithelial cells are ruled out because they don't have the CD genetics. And then they should have the morphology and functionality, which resemble the native tissue. And then also those models should be repeatable, reproducible. And then the culture and handling, because it's currently very laborious and labor intensive. It should be easily automated, and then the culture should be also miniaturized to have the high throughput analysis. For these current systems, the small intestinal organoids, which were already introduced by Arnold Han from the celiac disease patients, but also the induced pluripotent stem cells, which I'm soon going to represent, are very good. But then it would be also nice to have a possibility to generate an autologous multicellular model, meaning that you can have the whole tissue generated from the same patient cells, meaning that the whole genotype would be same for all the cells. For that, there is a brief presentation about what kind of autologous celiac preclinical gut chip could be. So there would be cells derived from induced pluripotent stem cells from the epithelial cells or the endothelial cells, and then they could be cultured together. Also, immune cells can be derived from iPSCs. Mainly currently the macrophages, but that is possible. And then we can add the environmental factors there, meaning nutrients or the microbiota, and then study the effects which are occurring on the epithelium. Since the invention of Yamanaka and Thomson groups, the iPSCs have been used very widely, but not in celiac disease. So the iPSCs are the cells which are derived from the somatic cells. Somatic cells are all our cells which are not the germline cells, so sperm and egg. And then the somatic cells which are differentiated, they can be induced to be pluripotent by introducing the pluripotency genes to them. And then thereafter, there is a characterization process to see that they are truly pluripotent and they are behaving as they should. And then after that, they can be differentiated and used to generate all different tissues and cells of the human body. The iPSCs of small intestines, intestinal epithelial cells, have been already differentiated for over a decade. Since 2011, Spence and others have been doing that, and then it is done it that way that the iPSCs are cultured within a jelly, matrigel, and then the cells form three-dimensional structures which have the intestinal kind of lumen, and then the cells resemble the human intestinal cells, so that they do have crypts and villi, and then they can be chopped and then passaged further. But then the culture like that, it has the apically inaccessible structure, which is not good for the, for example, the environmental effectors, triggers. And then the other thing is that, as Arnold already brought out, in the Galvin-Course paper, that they also retain the inflammatory cells, so that they also have. It is also pros and cons to have the inflammatory cells. If you want to study only the epithelial effect without yet having the inflammation there, then it's not a pros. The organoids can be also opened, so that they can be opened and then cultured on planar structures, so that the luminal part can be exposed easily to the environmental triggers. They can be on the left-hand side, as presented. They can be cultured on planar static culture, so that there is no flow, or on the right-hand side, which is the image from the Rahmani and others. Actually, this is from a Workman paper. They can be cultured on the chip model, where there is a flow of media on the cells, and then that pushes the cells to differentiate better. And then there can be also a stretching of the culture model, which also induces them to form the crypt and villi structure. That has also been done in the latest paper of Sebo Wichhoff and Iris Jonker's group in the Morkens and others, in reference in below. This culture system is extremely expensive, so that, it's really expensive. It's approximately three rounds is 10,000 euros or dollars. So that if you want to have 3 replicates, it's just the chip and the culture, and then you don't even have the device yet. So we wanted to overcome the 3D culture and then the opening of the structure. So we wanted to do the small intestinal model on planar, so that even when they were still pluripotent, we started growing them on flat surface. The Matsunaga and Michuguchi groups are the forerunners for developing those systems. But then we also wanted to optimize our iPSC culture with those methods. And I'll go that through. There is a scheme, so we have the iPSCs, which are pluripotent. Then we give certain cues for them to have the definitive endoderm, and then another set of cues you will hear at soon to get the posterior definitive endoderm, and then we have 21 days culture to get them small intestinal epithelial cells. When we did that work, we realized that having the fetal bovine serum, it inhibits the differentiation of cells. So on the right-hand side, you can see the TBXT, the brachyre, and then when you see the dip in the columns, that is the experiment where we did not have the FBS at all, and then we replaced the serum with the knockout serum replacement. And then the brachyre is the marker for the mesenchymal or mesodermal lineage, which we do not want to have because we don't want to have liver or kidney from our cell culture. We want to have intestine. We also showed that the xenophrene matrix it also supports the differentiation towards the definitive endoderm. So the Geltrex, which is an animal-derived factor, which is similar to the matrigel, is as good or not as good, or almost similar as recombinant laminin. So that with the recombinant laminin, we know what it does contain or what it doesn't contain. So it's easier to handle. We also showed that the xenophrene reducer, the LY thing on the left, it actually pushes the cells to differentiate better towards the posterior definitive endoderm than the recombinant BFGF, which is inducing several other differentiation routes than the LY. In the end stage, for the 21 days, we saw that the culture on gel tracks and also the laminin, they both result morphologically quite similar epithelial cells. And what we saw that they express the PEPT1 and vilin, and with the in the middle you can see that the laminin is actually inducing better expression with the vilin gene. Then we've moved forward to the celiac disease differentiation. This is now unpublished, so that we are still ongoing the data analysis and other things, so that all what I'm saying thereafter, we still need to verify, so that these data might still not be exactly true. But what we saw that without any stimulations, the induced pluripotent stem cells differentiate to small intestinal epithelial cells. In celiac disease patients, they already express CDXC10, which is usually induced by, for example, interferon gamma, which might suggest that the celiac disease patient induced pluripotent stem cell derived small intestinal epithelial cells have some kind of intrinsic behavior that they already express the CDXA10. And when we compared this in the middle, on the left-hand side, there is celiac disease. In the middle, there is type 1, and then on the right-hand side, type 2 cells compared to the healthy control. We don't see such effect, but we have to see whether that is true, because at least I'm a bit hesitant to say that that is true. 2. And then when we had the cytokine stimuli for the celiac disease patient cells, we see that there is induction of the genes. Actually, I can't see the text. The text is down there, is missing. Well, anyhow, there is IFIN 27 STAT1, and then CXCL10 induced by the interferon gamma, which are very well known genes which are induced by the interferon gamma. But the morphology of the cells appears to be normal even after 48-hour induction. And then we have new cell lines generated with the support of Beyond Celiac, and they look pluripotent and they express pluripotency markers, and they don't have the exogenous genes anymore, so that we are almost ready to use them. There is no text on this one. Well, as a summary, you can see only beautiful greetings from our celiac disease intestinal iPSC cell colony there, but then, yeah, and then greetings, the acknowledgements. This has been done in Tampere University Celiac Disease Research Center with the help of heart group induced pluripotent stem cell generation data analysis with the computational biology group, and then Folkhälsan, and then there are those supporters like Beyond Celiac and Cultural Foundation of Finland and Tampere Institute of Advanced Science as the major ones. And then you can see my little group. And then I welcome you all to the Tampere Celiac Disease Symposium 25. Thank you.
Thank you. Okay, I'd like to bring up now Richard Flavell from Yale University in Connecticut. His talk is entitled Generation and Validation of a New Humanized Mouse Model, a Surrogate for Human Celiac Disease. And then after his talk, we'll have some questions and answers.
Okay, and I just have to do that. Okay, thanks very much for the invitation. And I see the slide didn't do anything, so let's try.
No, it will.
It will? There it is. I went backwards. I could have done that for a long time. I could have redone your talk. So thanks very much. It's a great pleasure to be here and to meet both colleagues and patients, because it's a tremendously helpful reminder about what we're doing and why we're doing it. That's why I'm actually glad we work next to a hospital, because that forces one to remember those things. So I did it.
Welcome to conference.
There you go. Yeah. Well, if anyone wants to see anything from the previous talk, here we go. Okay, so what we've been doing for the last, since 2005, is to try and make small animal models of human diseases. And the fact of the matter is that humans and mice have diverged in evolution for something like, if you add it all up, 150 million years. So it's not surprising that if you try and model something in a mouse without any changes, that you get a lot of different kinds of results. And our goal is to make a mouse as similar to the human patient as is possible. Now, you all know that this disease is disaster for the world. It's all over the place, and it's despite understanding what the trigger is, it's extremely hard to manage. And it's basically, as you've heard already, it's an autoimmune disease driven by the trigger of being a dietary antigen and so on and so on. And I won't dwell on this because several people have covered it already. So I think everyone would agree that we need better mouse models of this human disease, just in general and in specific for this disease. And what do we want of those models? We want them to be responsive to gluten. We want the tissue, which is the duodenum, to be inflamed as a result. We want the tissue itself to respond in the way it does in people, which is the blunting of the duodenal villi. And we want the consequences of that to be manifested, which is poor absorption of food, malabsorption, in other words. And so we said about 3-4 years ago, Marie Robert, who is a pathologist at Yale, and many of you know her because she used to work with you. She is a very energetic person, and she kind of got a whole group of us together across the university and said, we've got to work on this disease and do something about it. And so thanks to her energy and enthusiasm, we've done that for the last few years. I'll show you all the team at the end of the talk, but it's been really exciting from my point of view to be involved in this. And so the goal, of course, is what I've just shown you. Now, the study of human disease has been made very much easier during this era of the multi-omics. You've heard proteomics, you've heard transcriptomics, etc. And so what we try to do is to add one feature to that, which is you can observe patients, you can observe the genes and the proteins of the patients, but what you can't do in a human patient is to ask, is this going from A to B? Is that the problem? And if it's true, if that's the problem, then what you have to do in a test, in a model, is to stop A going to B and ask the question, does the disease stop? You can't do that in a patient other than in a clinical trial, but you can do that in an animal model of this disease. And so that's what we're trying to do. That's what they call perturbations in vivo. So our goal is to determine cause and effect relationships, that something causes celiac disease, and then that's a prelude to stopping it, all right? Okay, so this is now samples of biopsies from our own patients that we look at. I'll explain who does what in the end of this talk. And of course, you see this very well-known epithelial blunting, which is the consequence of the disease. We use single-cell omics, of course, in order to get a roadmap for what's going on in the mechanism of this disease. So we start by looking at human tissue because we need to have a human basis to know that our mouse is doing the same thing. And so that, of course, involves histology of the patients. That's where Marie gets involved. Single-cell transcriptomics, which you've heard from several people already. And now more recently, spatial transcriptomics, because we want to know where in the villus, where in that intestine is the action happening? Where are the cells? Which cells are talking to which cells? And which cells are damaging which cells? That can be greatly helped by what we call spatial, which means you take a piece of tissue and then you determine where all the genes are, basically. So this is an example of a single cell experiment. You've seen many of these already in this section, so I won't dwell too much on it. This is comparing one of our celiac patients and a healthy control. And again, what's very clear from that, as you've heard already, is that there are major changes in the enterocytes, meaning the cells of the epithelium, of the gut epithelium in the duodenum. And also, as you see in the bottom reddish area, they're activated CD8 T-cells and gamma delta T-cells. So that's the scenario in the patient. So how do we get from that scenario to a mouse which will behave like that patient? Well, we make what are called humanized mice, and what we do is two things. We take human cells, because we don't want to be studying mouse cells. We want to be studying what human cells are doing. And we take those cells from a person that is susceptible for celiac disease. We then transfer them into a mouse, which is a good host for human cells. And that's the second part. So that you see number one, that's the human hematopoietic, the blood cell progenitors, in other words. I'll get back to that. The second point is to transplant them into a genetically modified mouse that has the factors that the human blood cells need in order to become immune cells. So it's very straightforward in concept, and basically we've been doing this since 2005. We do it in a specific way, which is that humanized mouse under number 2. What we do is take genes for human factors which we know are needed to make the immune cells from a human precursor in a mouse. Then we take that gene, and we only have to do this once. We take that DNA, we put it into the mouse genome, and we kick out the mouse gene, the corresponding mouse gene. So I'll give you an example on the next slide. So I went backwards, didn't I? I'm really good at going backwards here. Okay, so this is the mouse that we ended up with. And it has, on this slide, we have 6 genetic changes. So let's look at the top one, MCSF. This is a factor which enables an immune cell precursor, so a cell coming from the bone marrow, to become what we call a macrophage. Those macrophages are the cells that clear up debris. They're the cells that are always in the gut epithelium, and they are critical cells. So we just took the gene for human MCSF, we engineered it, so we put it into the mouse, and we threw out the mouse gene. You only have to do that once, because then you just breed the mice. And we did it with all of those genes. And what that does together is creates a mouse whose immune system is basically able, its immune environment is able to develop human cells. Now that mouse doesn't have human cells in it unless we stick them in it, right? So we do that. So that's this part that I just mentioned. So that mouse is now engineered, and we look at the top of that under number 1. We take human hematopoietic stem and progenitor cells. So these are the cells that make all blood cells, and we just transplant them into a baby mouse. You just take a needle, take the cells, it's not complicated, put them into the mouse, and they'll grow up in the mouse, and that mouse will have human cells, and you can study anything you want in those mice. Okay. So if you do that, we get a comprehensive immune system, and we've been doing that since 2005, and we've taken this system and shown systematically that it works. So the next step was, of course, to adapt this to celiac. I'm going to skip that. Just before I go into that, you can say this is looking at the tissues, and the most important point is in the gut of these mice, that happens to be the colon there, but it's relevant to the duodenum as well, which is the immune cells are in the right place in those mice. Okay, we can skip that. So because I think we're going to run out of time. Okay, so we built on that mouse, and we now need to make changes that are relevant to having a celiac disease model. So what are those changes? Conceptually, our mouse, which we call Mr. G, by the way, it's a cool name, isn't it? It wasn't anything to do with me. I had a postdoc, Choran Rathiman, and he took all the names of these genes and he lined them up and he said, we should call it Mr. G. And he's right. I mean, it was brilliant. His biggest contribution, I suspect. But it stuck, okay? So we still use it. He's the guy that did the MCSF, and so he did very important things. Anyway, so what do we do? So we've got to combine things that matter for celiac. So the first thing is a molecule called human IL-15. That is required in the intestinal epithelium to get the cells that you heard about in the first talk of this session, which are the so-called IELs. You heard about IELs. Those are the cells that live in that loop you see in the middle there, where you see those dots. I'm sorry, there's no cursor here, but I can show it. And that one, I think, did that do it? No, this thing. Forget it. You know, the problem is some of these screens don't respond to the laser, so that's what it is. Anyway, that gives us the cells which are in the epithelium, and we published that a few years back that works. So that's the first thing we do. The second thing we have to do, look under number 2, the human interferon gamma receptor. You've heard a lot about interferon gamma. It's made by the T cells and it causes the damage. Thank you very much. What do I point with? Laser pointer, not advancer. Okay. This thing here, right? Got it. Thank you very much. This thing, interferon gamma receptor, this is to enable the human interferon that comes from the human. Remember, they got human T cells from a person, but it's got to cause the damage to the engineered mouse gut cells, because at the moment we're using a mouse that has mouse intestine, human T cells. So we add this gene. We just pop that one in also. Now, if you treat that mouse with interferon, all this other stuff gets switched on, and it's human interferon. And we also get rid of the mouse ones. We don't want to have confusing mouse things going on. And then the third thing we have to add is all of the molecules which are required on the epithelial cell for a human immune cell, a human T cell, to get activated. And those are what you heard about. These are so-called MHC genes, HLA-B. This is the susceptible MHC class 1 gene that CD8 cells react to and NK cells look at. This is HLA-DQ 2.5, you've heard about already, so that we engineer that to be present on. And then another HLA gene, which is a so-called non-typical one, HLA-E. This is also implicated in this disease, and we also put that there. So we've done all of that stuff, and we've made mice that now have an engineered gut system that will respond to a human T cell. So this new system, that's a lot of things to say, and I'm going to use the next slide to say that, because that's going to drive you nuts, right? And we're at time, so that's perfect. Okay, so this is what we're ending up with, right? We can have a surrogate model of celiac disease in which we put into that mouse tissue from human, which is a thymus, a piece of thymus, and the immune cells from the same individual. That then grows up, those immune cells grow up, that person has exactly the same susceptibility genes for the T cells. It's DQ 2.5, HLA-B, et cetera, all that stuff. So we put that in there, and that mouse grows up with the immune cells from a susceptible environment, so-called genetically susceptible T cells. We then feed that mouse. Remember, that mouse has now got all those things on the epithelium we put there, right? So all those genes on when I say epithelium, it's the duodenum, right? That's the thing that's going to get damaged. So they're all there. So now we feed the mouse with gluten, and we monitor what's going on in the intestine. And so we've seen certain things. I'm going to skip this really. This is just a detail which is important, but we'll skip that. But basically, this shows that the IL-15 works. You have IELs if you want. If our mice that have IL-15 have IELs. If you don't have IL-15, they don't. HLA-B expression is well, this is a person that's induced, of course. This is showing from our biopsies and our single-cell sequencing. And our Mr. G mice, if you give them interferon gamma, remember we gave them the receptor, it induces the expression now of the molecule. So these tools are working in that mouse. Never mind the details, but hopefully the principles are there. So last slide, where do we stand with this whole thing? So we've made the mice which have IL-15. We've incorporated that into Mr. G. I just showed you the result. It works, basically. Next. We've put in the gene for the interferon gamma receptor. That's been made. We have the mouse, and it looks like it's working. We've made the genes for all of these guys, and we've put those into the mice at various times. We're still working on this one, but we're almost there. So we almost have this model ready to rock, as they say. So to summarize, where we're going now is to take this model to go through the manipulations required to test celiac disease, to give it gluten, to reduction in what they call reductionist, right? We're taking something complicated, simplifying it step by step, and hopefully we'll be able to figure out what's going on here.
Yep. Thank you very much. So we have time for some questions, and I'm going to ask the first one while people make it to the front. So this symposium is, of course, all about collaborations, and I'm wondering, how do you see the organoid work transferable to the mouse model?
Oh, that question for me?
Well, sure.
No, it is interesting. I think that, I think that we're kind of two ways to answer similar questions. I mean, we're more trying to use the organoids to dissect mechanisms, but I think that, what we learn from the mouse, obviously we can apply to ask questions in the organoids and vice versa, I think. I don't think there's a direct way necessarily to, for instance, put organoids into mice. I've thought about that, but I don't know, maybe in the future, but I see that more as different ways of answering similar questions.
I could comment. The next step for this model in general for us has been to introduce human organs into the mouse. We've done that successfully for the liver and the lung. And so we have human liver, for example, that functions like a human liver. That never happened before because you need multiple cell types of the liver to work. And we had a paper in Cell last year which shows that. In the lung, we've been able to do that. And we are planning to try, actually, to add human cells. We've done a couple of little experiments. It's going to be not trivial, but imagine what you have to do. You have to take these organoids, and you have to introduce them into the intestine of the mouse in a way that those cells will take over from the mouse cells. So you have to do two things, really. You have to put the mouse cells at a disadvantage because they've got to make space, and the human cells have got to take over. So this is one of the things we're trying to do, and we haven't done it yet, but I think in the years to come, this will be technically possible. But I'm not trying to trivialize it. I mean, your point's a very good point. This is not simple stuff.
Kati?
Well, I'm a cell biologist, and I'm pro 3R, which is reduction of animal use. So I'm sorry. I think that they are important, but then I think that with simplified models we can go certain steps, but then and then also I'm participating the organ on chip and then microphysiological system generation things. So I would rather see that the models which have human genes are important. We already know what happened with the thalidomide or with aspirin. So aspirin is very, very difficult for the mice, but then we tolerate it very well. So there are quite striking differences with our system. So this is just my opinion, but then in our institute we have very big group of people who are with the 3R and then pushing that also on the governmental level forward.
Okay, thank you.
I've just emphasized that it's all of these approaches are valuable, and one can learn a lot from cells on a chip, but whole organismal biology one cannot yet recapitulate. As soon as that can be done, then no animals will be needed ever, actually. But we haven't really reached that stage yet. And of course, the other thing that's going to happen is eventually this field, as all fields, will become data. We won't need experiments, actually, or only very, very few experiments. There are already 250 single-cell data sets of human out there that you can analyze everyone, and in the room, you get a computer kit to work with you. They can do all that analysis. It's a wonderful world we're living in at the moment. Well, not world. The world's not so wonderful, but science is wonderful.
Science is wonderful. Yes.
Sorry.
Amélie Terrien, Boston. I have a question for Dr. Han about double negative gamma T cells. Did you characterize them in your recent analysis? What do you think they do?
No, I don't think we care. What do you mean by double negative like that?
CD4, CD8 negative gamma delta T cells.
We didn't really look at CD8 on them. I think that there's been a question about whether the gamma deltas in celiac disease have CD8 or not. And, at least based on my prior work, there isn't any clear pattern. Like some patients, they mount CD8 positive gamma deltas, and other patients mount CD8 negative gamma deltas. So I think that, and phenotypically, they're more or less indistinguishable. So I'm not sure about the role that CD8 actually has.
And I have a question for Dr. Flavell. At first, you saw a figure with spatial transcriptomics. Was it published?
No. Everything I showed on celiac disease is unpublished.
Okay.
Not a single thing is published.
Okay.
So that's easy.
No, yeah, because I'm interested in doing that. Anyway,
Yes,
I can
that
chat
was
with
done
you
with
later.
the COSMX
Okay.
system. Yeah.
Thank you.
And that was a UMAP plot next to it. There was no time, right? But that was analysis of the genes and their clustering. And the colors mean the color on the tissue, which is the middle section, corresponds to the colors on the UMAP plots. You can see which genes are where.
Thank you.
Sorry about that.
Yeah, I mean, this whole conference is all about collaboration, so please talk to the investigators after. Yes, one last question.
Hi, sorry, my name is Grace. I have a question for Dr. Han. So I do research on mesenchymal stem cells, and I've seen that they can reduce the CD8 T-cell expressions. So do you see any potential in correlating the T-cell modulation with mesenchymal stem cells, or just any stem cell in general?
Interesting question. Tell me a little bit more about the research that you've described or what you figured out.
So I use mesenchymal stem cells derived from human deciduous teeth because my sister has a genetic predisposition. So it's like a non-invasive way of obtaining the stem cell. And then I inject them into mice that I've given celiac disease to, and then I just do the research from there, and I've seen success, I guess. Sorry, I don't know too much of the scientific terms to
So you're injecting in what kind of mouse are you injecting in?
Rag1 mice.
Oh, and what do you see in the CD8 cells?
A reduction in them.
Okay. Yeah, no, I can't really comment on how that relates to human disease because we haven't really looked or addressed that particular question. I mean, clearly it's not just immune cells, we think. Our data does show that there are differences in the epithelium as well. And I think that those epithelial differences are mediated actually by immune cells. But I think that's an interesting question as to, for instance, what distinguishes these potential celiac patients from regular celiac patients. We see that there's something clearly different in their epithelial cells. So non-immune cells, but I'm not sure if I've really answered your question, but you can come and talk to me later.
Yeah, maybe you two can talk. It sounds like you're doing really interesting research.
All right, thank you.
Okay, thank you very much, the panel. And we're going to take a 10-minute break, so until 11:25 on my watch at least, and we'll tell you when it's time to come back. So thank you.